The overall goal of this procedure is to generate primary orthotopic glioma xenografts to recapitulate histopathological and molecular features of malignant glioma subtypes in preclinical animal models. This is accomplished by first obtaining human brain tumor specimens for direct transplantation. The second step is to prepare a cell suspension from the specimen. Next, the tumor cells are transplanted into the brain of an immunocompromised mouse. The recipient mouse is then monitored for signs of tumor engraftment and growth, and then harvested for analysis or serial transplantation. Ultimately, histology, DNA sequencing, and other molecular assays are used to show infiltrative tumor growth and retention of genetic alterations such as isocitrate dehydrogenase mutations.
Preclinical animal models of cancer are essential for investigating tumor biology and therapies. The main advantage of primary glioma xenografts over existing methods such as secondary glioma xenografts is that glioma cells undergo genotypic and phenotypic changes in culture that cannot be restored when cultured glioma cells are transplanted into mice. Demonstrating the procedure with me today will be Dr. Geraldo Valadez, a member of my laboratory.
To begin, prepare the papain dissociation solutions in a sterile hood with the kit components and instructions